The field of psychology, specifically neuroscience, explains a lot of phenomena related to our behavior and the biological causes. One of these phenomena is what we call curiosity. Curiosity is the driving force in childhood development, the key to educational attainment, and a major propeller for scientific discovery, which pushes our society forward. The part of the brain that has been proven to be most responsible for curious behavior is in the hippocampus, called the dentate gyrus. The functions of the dentate gyrus include adult neurogenesis, which is the generation of neurons throughout one's adult life, the formation of new episodic memories, and the spontaneous exploration of novel environments, which, when simply put, is curiosity. An experiment was done involving proteins in the dentate gyrus. Researchers studied the effect of the protein NCS1, neuronal calcium sensor 1, in a group of mice. This protein was already known for its ability to affect memory in worms, and it's linked to bipolar disorder and schizophrenia. They increased the concentration of the protein 1.5-fold in the dentate gyruses of the experimental group of mice and compared the effects with the untreated group. The treated mice expressed superior memory in complex tasks and increased curiosity. Aside from the dentate gyrus, other structures play a role in curiosity as well. The prefrontal cortex houses our working memory. This allows us to distinguish between new and previously experienced stimuli, which in turn keeps our curiosity under control. A part of the brain that may play an even more important role is the reward pathway. A rewarding stimulus triggers a signal that travels from the ventral tegmental area to the nucleus accumbens to the prefrontal cortex, which we mentioned before. When we actively pursue the knowledge of unknown information, we are rewarded with a temporary flood of the neurotransmitter dopamine, which regulates movement, learning, emotion, and most importantly in this case, pleasure and reward. Here's a summary of how neurotransmitters work. First, an action potential is triggered in a neuron, which is when the membrane potential of the neuron fluctuates to allow for depolarization, repolarization, and the refractory period. This occurs down the axon until it reaches the axon terminals, where vesicles containing the neurotransmitters disintegrate with the help of calcium, and neurotransmitters leave the presynaptic neuron and enter the synapse to bind to the receptors on the dendrites of the postsynaptic neuron. This triggers an action potential and the sequence starts all over again. By turning some of our focus in the field of psychology, specifically neuroscience, to how curiosity works in the brain, we can further discover its effects in all kinds of species, such as humans, mice, and more. We can then apply this information to how learning, behavior, and developmental processes work in animals and humans, which could lead to treatments, medications, and therapies that could aid people who suffer from conditions within this category. Curiosity did kill the cat, but the research could also cure it.